All right, so today we're going to be talking about Western blotting. So, what is it, how it's conducted, and a little bit about how to interpret it. So, let's talk a little bit about what we can use a Western blot for. So, a Western blot is basically a technique that scientists can use to determine the relative level of proteins in a cell of interest. The Western blot can be used to separate proteins by size or semi-quantitatively. And as you can see in the rat tissue panel on the bottom of the screen on the right, we can determine the relative level of protein in a sample by looking at the strength of the band. So based on this, we can see that the lung tissue has the highest beta-actin expression because it is the brightest band in the row. We can also use the band to make comparison between samples. So just to give you an example, we can tell that there is more beta-actin in the lung tissue than in the liver tissue because the lung band is brighter and larger. This is a very useful technique in research because it allows scientists to measure protein content in a cell. So just to give you a few more examples of CJD, which is a fatal brain disorder, can only be identified in humans by running a Western blot on a patient sample. This is also true for HIV and AIDS, as an anti-HIV antibody can be used, which shows if HIV is present in the cells. More recently, the Western blot has been used to test for blood doping. So for those of you who don't know, blood doping increases the number of red blood cells in the body, increasing athletic performance and endurance. This has been a major problem in sports recently. You might have heard of this in relationship to bicycling. And the Western blot is currently the World Anti-Doping Agency's official test for blood doping in athletes, and was used to test every athlete that participates in the World Cup starting in 2014. So, now we know a little bit about why you would use a Western blot. So how does one actually conduct a Western blot? Well, there's two basic steps. So the first is the SDS page step. When you run an SDS page, proteins travel based on size, with larger proteins staying closer to the top. So SDS stands for sodium dodecyl sulfate, which is a solution that denatures proteins and eliminates factors that may influence how they travel, except for size. The samples are then run using PAGE, which stands for polyacrylamide gel electrophoresis, which is the physical running of the gel itself. The second basic step is immunoblotting, which involves using a pair of antibodies to bind to and detect a protein of interest. A Western blot is an extension of SDS page because you use the SDS page and then transfer it to a membrane with an antibody. There are a few other steps involved in this, which will be talked about more in detail on the next few slides, but to give you a general overview, the first step would involve sample preparation, and samples come from cells or tissues of interest, so whichever kind of tissue you want to isolate a protein from. Protein samples are then run on the polyacrylamide gel with SDS page to denature the samples. The gel is transferred to a membrane and then can be stained with a primary antibody to label the protein of interest. A secondary antibody is also added that binds to the primary antibody and is fluorescently labeled. The fluorescence can then be detected, allowing the protein to be visualized, and all of these steps will be talked about a little bit more in detail on the next slide.
So I'm going to take you into a little more in-depth on these 5 steps that Elizabeth had just went over. Step 1 is your sample preparation and your gel electrophoresis. Your source of proteins will come from cells or tissues which you are targeting. The proteins of these cells will be loaded in equal amounts into the wells of an SDS page gel along with a ladder. The ladder is also your molecular weight, and this will be made ahead of time. Usually you want about a 4 to 20% gradient gel. The gel will be ran for approximately 5 minutes at 50 volts, but this can vary depending on how fast you want to finish. This step will separate the various proteins of interest. Step 2, transferring to a membrane. After the gel has been ran, it will be placed in a transfer buffer where it will be made into a sandwich between the blot, which is a nitrocellulose sheet, and the container. An ice block is then added to the tank to keep the temperature low, and the tank is then transferred to a cold room where it will have a constant current of 10 milliamps ran through it. On to the third step, immunoblotting. The membrane will be washed with and blocked with a nonspecific protein such as TBST. This nonspecific protein will bind to the membrane and prevent any antibodies from binding to the membrane. The primary antibody will then be added to the solution which the membrane is in. After a wash to remove unbound primary antibodies, a secondary antibody is added along with the enzyme. There will be a second wash conducted after this to remove the secondary antibodies. The fourth step, immunodetection, is where detection is done through an enzyme that was added with a secondary antibody that is paired with a chemiluminescent substrate. Following the binding of an enzyme and substrate to the membrane, an emitted light that can be detected with a film or photoimager is used to visualize the bands. The fifth and final step, which is the detection step, is where the film must be lined up with the membrane in the exact same manner that it was exposed, because the film does not tell us where the lit bands are located in relation to the membrane. After this step, the membrane will be stained so the protein can be visualized and compared with the film.
So for chemiluminescence, I kind of like this picture because it describes everything. So Western blots are probed with a primary antibody against the target protein, followed by a secondary antibody labeled with the HRP, which stands for horseradish peroxidase enzymes. In general, the primary antibody that recognizes the target protein in a Western blot is not directly detectable. Therefore, tagged secondary antibodies are used as the means of ultimately detecting the target antigen, which known as indirect detection. HRP is the most commonly used reported molecule for secondary antibody in a Western blot. A chemiluminescence substrate for the HRP enzymes is carefully applied to the blot. HRP will react with the hydrogen peroxide. Then light is emitted when the HRP enzyme modifies the substrate. Photographic film or an imaging system using a digital CCD camera captures the emitted light as an image.
So there are many advantages to using a Western blot over other techniques. Western blotting gives you the ability to isolate a specific protein of interest and is capable of detecting multiple proteins at the same time. It allows researchers to obtain all the results for what they are interested in without having to sift through extra data. The results from a Western blot are easily comparative to one another and can be readily reproduced to help confirm results, allowing this test to be double-checked for accuracy. And while there are several steps to the Western blot, it is relatively easy to conduct this, primarily due to the fact that most of the equipment used in the technique is common laboratory equipment and needs no special skills.
So in recent years, there have been several advances in Western blotting. The first is capillary or microfluidic platform, or CMP. The CMP have several benefits. First is that it takes less time to conduct. It allows for analysis of protein content down to a single cell level, which is highly specific. Most importantly, a significantly smaller amount of sample is needed to run the Western blot, allowing for more precision. Another huge advance in Western blotting is the use of a charge-coupled device, or CCD, which can detect both fluorescence and chemiluminescence in a sample altogether. This, coupled with enhanced chemiluminescence detecting agents that are more sensitive and needed in a smaller amount, provides a more reproducible experiment that is much more sensitive to the amount of protein in the sample. And these are our sources.
Thanks.